Hello students, hope you all are fine and doing well. So welcome to my channel. Welcome to Dr. Sandipan Sengupta's Modern Biology meant for degree courses and various competitive examinations like CSIR-NET, GET Life Sciences, NET examination, IIT-JAM, etc. So you are listening to your very favorite Dr. Sandipan Sengupta, a University Assistant Professor of Microbiology and Biotechnology. Today, I'm over here with a new lecture session of microbiology in which we'll be discussing about the history and development of microbiology. It's going to be a very thorough lecture session in which we'll go through each and every major development corresponding to the major eras of microbiology, that is discovery era, transition period, golden era of microbiology, and the era of modern microbiology or molecular microbiology. And all these things, that is, all these discussions, that is, would help you to write up your BSc and MSc degree course examinations, and a lot of questions are frequently being asked in GATE Life Science Microbiology examination pertaining to the sections which we are going to discuss today. So stay tuned, and after listening to this entire talk, if you still have doubts and queries about the history and development of microbiology, do leave your mail ID at the comment box, in the comment box, and you'll receive a free PDF of the topic. But you can only do this after you are listening to this entire talk without skipping any segment. So before putting up or coming up with your queries, and before putting your mail ID in the comment box to receive a free PDF or free note of today's topic, be sure that you are listening to the entire talk very thoroughly without skipping any segment. So this is going to be a very engaging and very thrilling and exciting lecture session for you all. So let's start. So let's take an introductory look over microbiology. So what do we mean by microbiology, and what are microorganisms? Which organisms can be classified as microorganisms? Microbiology involves the study of small microscopic organisms which cannot be observed under a naked eye and which need to be observed or which can be observed only under a microscope. A microscope magnifies the images of these microscopic or small organisms, and then only they can be observed. But what is the reason behind this? Why is it that these organisms cannot be observed under a naked eye? This is because of the small cellular dimensions of the organisms, so-called microorganisms. So the group includes only those organisms which have or which are having a cellular dimension of 1 millimeter or less. So such organisms are classified and known as microorganisms. So there are a wide and vast range of microorganisms, and the most prominent one the most prominent one being bacteria, protozoa, algae, fungi, helminths and some worms, and viruses. So the discipline involves particularly the study of bacteria, only microbes which have a cellular dimension of less than 1 millimeter. And the most important ones included classified as microorganisms include bacteria, protozoa, algae, fungi, helminths and some worms, and viruses. So among all these, only bacteria have a prokaryotic mode of cellular organization, while algae, fungi, protozoa, the helminths all have a eukaryotic mode of cellular organization. And viruses, when we speak of viruses, they are unique in having acellular mode of organization. Viruses have been structured around a nucleocapsid core, often surrounded by an envelope. And they also have a unique pattern or unique kind of life cycle because they are considered as living only in their intracellular phase of life cycle when they are present inside host pathogens, and when they are present outside the host cells, that is in the cellular environment, they are considered as non-living. Now, let's directly move on to the topic. We'll see the major developments that took place in each and every era included or landmarked in the history of microbiology. So the history of microbiology can be divided into 4 different era, or major developments took place in 4 different, that is, distinguished periods, that is referred to as the discovery era, the transition period, the golden era of microbiology, and the era of modern microbiology or molecular microbiology. So presently, we are going through the era of modern microbiology or molecular microbiology, and a lot of advancements have taken place in the discipline, all of which have paved the way, and as a result of which it arose as an independent discipline. And presently we are going through the era or the epoch of modern, or we can even say extra modern or ultra modern and very scientific molecular microbiology. Now let's start with that is developments that took place in the discovery era. So the discovery era actually, that is, deals or only included the discovery of microorganisms, and the discovery era was dominated by the contributions only or exclusively made by Antony van Leeuwenhoek. So what were the contributions of Antony van Leeuwenhoek, and how were these microbes discovered? So Antony van Leeuwenhoek was the first biologist who could precisely observe and minutely or finely describe small microscopic particles, which he called as animalcules. And later, these animalcules discovered by Antony van Leeuwenhoek were found to be a collection of bacteria and protozoa. Antony van Leeuwenhoek, by 1665, had constructed around 250 small microscopes which could magnify in between 50 to 300 times. So by such an early time, or by such an early, that is, age, now Antony van Leeuwenhoek, using his own or self-constructed microscope, was the first to observe and give or come up with a precise description of microorganisms, so-called as animalcules, which were nothing but a collection of bacteria and protozoa. So because of immense contributions which Antony van Leeuwenhoek made in the field of microbiology, he is considered as the father of bacteriology and protozoology. Now, here's a view of the small creatures called animalcules, which were observed and discovered for the first time as microorganisms by Antony van Leeuwenhoek. And these were nothing but a collection of bacteria and protozoa. And for this reason, Antony van Leeuwenhoek is considered as the father of bacteriology and protozoology. So with this ended the discovery era of microbiology and began the transition period of microbiology. So what were the developments pertaining to the transition period? Let's have a look. Now, the transition period, the major developments that took place in the transition period were all focused around one major issue, that is the controversy of spontaneous generation or abiogenesis over biogenesis or versus biogenesis. So the transition era of microbiology involves the work of two group of that is biologists, that is one group of biologists, one included biologists who were in favor of the theory of abiogenesis or spontaneous generation, and the second group included other biologists, all of that is who were in favor or support of biogenesis. That is, and the biologists favoring or supporting spontaneous generation or abiogenesis had their own explanation about the origin of microbial life, while the other biologists supporting the doctrine of biogenesis had a very scientific explanation behind that is working as the reason behind the origin of microbial life. So what was the theory of spontaneous generation or abiogenesis, and what do we mean by biogenesis? Let's have an individual look over these terms first. Now, by abiogenesis or by spontaneous generation, we mean to refer to the fact that life arises from inanimate matter or non-living object. So it is the vital force of the vegetative source of nutrients present in food that could give rise to new life or to new microbial world. So the microbes were thought to arise spontaneously as a result from inanimate objects or non-living objects, and so such was the theory of spontaneous generation or abiogenesis. That is, there were a group of biologists who were in favor of this, in support, who conducted experiments in support of this theory of spontaneous generation or abiogenesis, and the most prominent or among them was John Needham. While all other biologists and most of the biologists of transition period were in favor of biogenesis or theory or doctrine of biogenesis. So by the doctrine of biogenesis states that life arises only from pre-existing life as a result of multiplication and cell division. That is, what is the source of microbial life according to the doctrine of biogenesis? It is only pre-existing microbial life that can give rise to new microbes following the multiplication, reproduction, and cell division of the pre-existing microbes. So this was the doctrine of biogenesis, and most of the biologists conducted experiments in favor of, or in order to prove and support their own, that is, proposed doctrine of biogenesis. Now let's take a look, independent look over the experiment conducted by each and every biologist who, that is, in favor of abiogenesis or biogenesis in the transition period. Now, there were four major biologists who were actually, that is, working over the issue or over the controversy of spontaneous generation or abiogenesis versus biogenesis. Now, one was Francesco Redi, another one was John Needham, then the third one was an Italian biologist or naturalist called Spallanzani, and the fourth and the most prominent one was Pasteur. So among these four, that is Francesco Redi, John Needham, then Spallanzani, and Pasteur, it was only John Needham who was in favor of, or who supported, the theory of abiogenesis or spontaneous generation, while all other three, that is Francesco Redi, then Spallanzani, and Pasteur, were in favor of the doctrine of biogenesis and strongly believed that microbial life originates only from pre-existing microbial life following multiplication, reproduction, and cell division of already existing microbes. Now, let's have an individual look over experiments performed by these microbiologists to support their own theory. Now, Redi, Francesco Redi was the France was the first biologist who actually designed an experiment, and he was in favor or support of biogenesis. He disproved or dissupported spontaneous generation or the theory of abiogenesis by designing a very simple experiment in 1665. He took meat and placed that meat in three different jars. So the mouth of one jar was uncovered, and the meat in the first jar was totally accessible to outside microbial world, while the mouth of the second jar was covered with a silver gaze paper, and the mouth of the third jar was covered with a simple paper. Now, following incubation, that is, microbial growth could only be observed over the surface of the meat that was present in uncovered jar. So when Redi was asked to explain, Redi gave an explanation that flies assessed the meat which was present in the open jar. Flies landed over the meat present in the open jar, laid eggs, and those eggs developed into maggots. So maggots could only be observed at the end of the experiment in the open jar in which had uncovered meat, while the other two jars could not or did not show the presence of maggots. So Redi further concluded that maggots were nothing but the offsprings of flies, and it was only pre-existing life that assessed the meat present in the open jar, or that landed over the meat that is in the form of flies, laid eggs, and those eggs developed into maggots, and hence pre-existing life gave rise to new that is life. And thereby Francesco Redi proved his doctrine of biogenesis, and disproved the theory of spontaneous generation by conducting a very simple experiment. However, now he was soon refuted and challenged by a biologist named John Needham, who was a great supporter of abiogenesis or spontaneous generation. And John Needham was in fact the greatest supporter of spontaneous generation and was the only biologist to conduct experiment in favor of spontaneous generation or abiogenesis. So doesn't it sound very interesting? It's interesting indeed. Keep on listening to the experiments and the way these biologists, that is, were designing the experiments to prove their own doctrine. So what John Needham did in order to, that is, prove his theory of abiogenesis, John Needham showed that his boiled mutton gravy or sterilized mutton gravy kept inside a jar whose mouth was covered with a cork would show the presence of animal cues. Although the jar had sterilized mutton gravy and the mouth of the jar was covered with a cork, and the mutton gravy inside the jar was inaccessible to the outside microbial world, but still it could show the presence of animal cues. So John Needham, thereby based on his observation, concluded that or proved his doctrine of abiogenesis or spontaneous generation of life from inanimate matter or non-living objects. But what might have been the reason behind the appearance of animal cues? When further experiments were conducted, it was found that John Needham could not sterilize the mutton broth properly, and in thus, although he covered the mouth of the jar in which the mutton broth was kept with a cork, but still the mutton broth already had the microbes, pre-existing microbes, and already was associated with the pre-existing microbes that gave rise to animal cues. Now, John Needham's work then was soon refuted and challenged by an Italian biologist or naturalist named Spallanzani. Spallanzani was again a huge supporter of the theory of biogenesis. And Spallanzani designed his own experiment to prove that microbial life only arises from pre-existing microbial life. So what Spallanzani did, Spallanzani took beef broth, boiled it for a brief period of time, and sealed it inside a small container or a glass jar. Following incubation, no growth is observed, and there is no origin of microbial life in the glass jar, because the broth that is beef broth present in the jar was inaccessible to the outside microbial world. Now, but soon Spallanzani was challenged by Needham and was accused of destroying the vital force, so-called the vegetative source of the nutrients present in the beef broth, and was accused of boiling the beef broth for a huge longer period of time. So in order to that is address this challenge, what Spallanzani did, Spallanzani made a small crack or neck, a small hole in his flask, and then showed that heated, that is broth, could still show the presence of animalcules when a small crack was made in the neck and it was exposed to the outside microbial world. So the experiments are very interesting indeed. That is, and this was the way by which Spallanzani proved his doctrine of biogenesis. But still, people all were not that is equally convinced, and there were a lot of controversies circling around the two theories, that is about the origin of microbial life, and people were still confused that whether it was a biogenesis or spontaneous generation that was giving rise to microbial life, or it was biogenesis or pre-existing microbial life that was the source of new microbial life. So with these, that is queries, with these contradictions prevailing, ended the transition period of microbiology, and the controversy of spontaneous generation or abiogenesis over biogenesis or versus biogenesis was finally, well, that is, could not be finally resolved by neither Francesco Redi nor Spallanzani. Now, who was the biologist who could finally resolve the controversy? It was Louis Pasteur who could finally resolve the controversy of spontaneous generation over biogenesis, and Louis Pasteur who finally put the theory of spontaneous generation to rest and proved the doctrine of biogenesis, but that he did in the golden era of microbiology. So let's have a look over the developments that were taking place or that took place in the golden era of microbiology and the work of Louis Pasteur and one more microbiologist, Robert Koch. So when we speak of the golden era of microbiology, the golden era started in around 1860 and spanned for 5 decades, for 50 years, till 1910. So the golden era of microbiology was dominated by the contributions made by two major microbiologists, that is Louis Pasteur of France, and the second microbiologist was Robert Koch of Germany. So Louis Pasteur and Robert Koch made, that is, brought an important advance, and that is brought a series of inventions which marked the beginning and end of the golden era of microbiology and paved the way for the era of molecular microbiology or modern microbiology. microbiology. Now, the golden era began with the experiment of Louis Pasteur, which he designed or which he did conducted in order to put the theory of spontaneous generation to rest. So what Louis Pasteur did, he designed his own flask, a series of flasks which were called Swan necked flasks. Why were they called so? Because they had their necks curved and the necks were shaped as U-shaped tubes. So the flasks were so ingenious and so unique in their observation and design that they were given a unique name and were named as Swan necked flasks because they had U-shaped curved mouth openings. And in these, in those U-shaped curved mouth openings, dust particles carrying microbes or dust particles which were the source of microbes could easily settle down due to gravity. So now what Louis Pasteur did and how he conducted his experiment, he took nutrient solution and poured it in his Swan necked flask, boiled and sterilized the flask along with the nutrient solution for a brief period and incubated it. The mouth was left uncovered. Let's take a look. So Louis Pasteur filled his, one of his round bottom Swan necked flask with nutrient broth, heated the contents so that and sterilize the contents, and following sterilization or following which the flask along with the nutrient broth was subjected to incubation for a small or for a brief period of for a definite period of time. However, one interesting thing was, or observation which we can see over here, is that the mouth of the U-shaped Swan-necked Louis Pasteur's flask is uncovered, or was the mouth, Louis Pasteur kept the mouth of his flask open. The mouth was accessible to microorganisms, but following incubation, there was no growth over nutrient broth. And nutrient broth could not show the presence of new and the origin and presence of new animal cues. So when Pasteur was asked to explain his observation, Pasteur gave a very scientific explanation that the mouth was open, and through the mouth, now the dust particles bearing germs had tried to enter and had tried to assist the nutrient solution, but the dust particles were made to settle or had settled down in the U-shaped neck opening of the flask due to gravity. So while the dust particles settled to this U-shaped naked portion, that is the neck part of the flask, the microbes also settled down, and the microbes could not assess the sterile nutrient broth, and hence there was no microbial growth. And finally, in order to, as the next segment of his experiment, he broke the neck of the flask. When he broke the neck of the flask, the dust particle or carrying germs and microbes could easily assess the nutrient broth, and after a brief period of time, microbial life and animal cues could be observed in Pasteur's flask. So when the mouth was broken and the sterile nutrient, the medium in the flask was made accessible to outside microbial world, then animal cues could be microbial life originated in Swan-necked Pasteur's flask, and new microbes could be observed. So this in 1861, this experiment of Pasteur, so-called Swan-necked flask experiment, finally put the theory of spontaneous generation to rest, and Pasteur could prove his doctrine of biogenesis, and it was established. Pasteur had already established by his experiment that microbial life, originated from pre-existing microbial life following multiplication, reproduction, and cell division of pre-existing microbes. So the pre-existing life is the source of new life, or it is the doctrine or theory of biogenesis that forms that is base for the origin of microbial life. So this was the first contribution made by Louis Pasteur, but beyond and beside this, Pasteur kept on making a series of contributions, all of which became landmarks of the golden era of microbiology. Now let's have a look over other contributions of Louis Pasteur. There was a superstitious belief circling around there was a controversy or a superstitious belief circling around the fermentation of grape juice to wine. So people were actually believing that the conversion of grape juice to wine was a chemical process which involved the breakdown of a protein named albumin. So it was thought that the grape juice was getting converted into wine as a result of chemical breakdown of a protein called albumin. However, it was Louis Pasteur who disproved this and came up or concluded by his own experiments that the conversion of grape juice to wine is not a chemical but indeed a biochemical process that is brought about by a eukaryotic microbe, that is yeast, biologically named as Saccharomyces cerevisiae. And the process is named as or called as fermentation. So it was Louis Pasteur who could firstly introduce the concept of fermentation, that the conversion of grape juice to wine is a biochemical process, so-called fermentation, that is driven by the activity of a eukaryotic microbe or a yeast microbe, biologically called Saccharomyces cerevisiae. Now, this was the second major contribution that was made by Louis Pasteur. Next to this, he introduced the concept of pasteurization. So while studying the process of fermentation, and while that is observing studying the role of yeast, Saccharomyces cerevisiae, and in converting grape juice to wine via fermentation, Pasteur found that the microorganisms could also cause spoilage of wine, degrade the quality of wine, and microorganisms were capable of spoiling wine. So Louis Pasteur then, next to this, found that it was not only wine, but it was also milk that was frequently getting spoiled by the activity of microbes. So Pasteur found that milk was getting spoiled due to activity of microbes, and to prevent this, he developed and introduced a new concept or a method called pasteurization. So Pasteur suggested that is milk and milk products that is subjected to boiling, that is subjected to mild heating at temperature well below boiling, that is at 62.8 degrees Celsius, and such an heating can be continued for a brief period of time, say about 30 minutes, to destroy the unwanted microorganisms which would otherwise cause spoilage of milk. Hence, and this was the concept of pasteurization that was introduced by Louis Pasteur. And so the controlled heating of milk and milk products at temperatures well below boiling at 62.8 degrees Celsius for a brief period of 30 seconds can reduce the population of undesirable microorganisms present in milk without ruining or spoiling the taste of milk. So such was the concept of pasteurization introduced and by Louis Pasteur, and by 1892 pasteurization became commercially very popular and became a regular practice in each and every milk and milk based industry of USA and around the world. And nowadays pasteurization that is the process has gone through a series of changes, and a lot many techniques have been have been brought into practice, so called, that is, there have been a lot of modifications, and the process has been modified and to make it suitable for large production, that is industrial production, and to handle industrial production of milk and milk based products like bulk pasteurization, flask pasteurization, then high temperature short term pasteurization, etc. So Pasteur, these were the major contributions of Pasteur. He first brought the theory of spontaneous generation to rest and proved the doctrine of biogenesis. Secondly, he introduced the concept of fermentation. And thirdly, he introduced the concept of pasteurization. But still, his contributions are not over, and Pasteur's work was even not over. Louis Pasteur kept on studying about the role of microorganisms, and Pasteur gave a very sound explanation that microbes which could spoil wine, microbes which could spoil or which could deteriorate the quality of milk could also make human that is beings fall sick, could also make human organisms or humans fall sick. And this laid the corner or the foundation stone for Koch postulates and germ theory of disease that was invented by Robert Koch and became a major milestone development in the field of medical microbiology. So next to Louis Pasteur, it was Robert Koch who made a series of contributions, all of which have become record-breaking contributions concerning or pertaining to the golden era of microbiology. Now, let's take a look over the contributions made by Robert Koch of Germany in the golden era of microbiology. Hope you're enjoying the lecture session, and we are coming across the contribution of each and every microbiologist pertaining to each and every era of microbiology. Now, let's have a look, let's study a bit and know a bit, that is, know in detail about a contribution made by Robert Koch and the way he introduced germ theory of disease, and he introduced started as a result of which medical microbiology emerged as an independent discipline. So Robert Koch was the first who could prove the involvement of bacteria as a pathogen in causing diseases, so-called anthrax bacillus. So Koch isolated and discovered Bacillus anthracis, the causative agent of anthrax, and anthrax was Bacillus anthracis, the causative agent of anthrax, was the first pathogen which was to be discovered, which was discovered or invented by Robert Koch. Now, soon in 1882, he discovered another pathogen named Mycobacterium tuberculosis, which is the causative agent of a serious disease called tuberculosis. Now, another major contribution of Koch was perfecting the technique of making solid media using gelatin as a solidifying agent. So Koch popularized the use of gelatin as a solidifying agent to prepare solid media plates under laboratory conditions for growing microbes. But the most important and most noteworthy contribution of Robert Koch was his findings reported as the germ theory of disease. So the germ theory of disease actually consists of 4 different postulates, all of which were experimentally proved by Robert Koch, and the postulates are still in practice to investigate the etiology or the cause behind a specific disease, and postulates helped initially to lay, that is, to establish a relationship between the causal organism, so-called the pathogen, and the affected organism and or the diseased organism, so-called the host, in the field of medical microbiology. So these are the postulates of the germ theory of disease that are still in use to investigate the etiology or the cause behind each and every disease that is caused in the field of that is noted in the field of medical microbiology. Now let's have a look over the postulates one by one. The diseased organism, that is the suspected pathogen, should only be found in the diseased organism and not in the healthy one. So this was the first postulate, and Robert Koch, that is, collected specimens, that is 2 different specimens, one from a diseased individual and the second one from a healthy individual, and cultured the specimens under laboratory conditions and plated them. So following incubation, the plate of the disease specimen plate only showed the presence of the disease causing pathogen, while the healthy specimen plate did not show the presence of pathogen. And thus Koch concluded that the disease, and that is pathogen, should only or is only found in diseased organism but not in healthy individual. Now secondly, or next to this, Koch actually perfected the technique of isolating this disease causing pathogen in pure culture, which he called as axenic culture, from a mixed population under laboratory conditions. So what is the second postulate of germ theory? Which the pathogen, that is, it must be isolated under laboratory conditions as an axenic culture, or the disease causing pathogen has to be isolated under laboratory conditions from a mixed population in the form of axenic culture under laboratory conditions. Now the third, in the next, to prove his third postulate of the germ theory of disease, Koch introduced this isolated pathogen into into an experimental organism. And when the experimental organism was thoroughly examined, he was found to contract the same disease that is caused by the introduced pathogen. So this was the third postulate of the germ theory of disease, laid or postulated by Robert Koch, that when the isolated pathogen is introduced into a new healthy individual or into a new experimental organism, it should cause the same disease. And this was indeed experimentally proved by Robert Koch. Now, the last postulate of the germ theory of disease, which was postulated and which was put forward by Koch, was that when the diseased experimental organism is examined and is looked up for the pathogen, the same pathogen should be isolated from the experimentally infected organism. So Koch, through his experiments, again proved, and when the experimented organism, that is, which was actually made to contract the disease by the introduction of the isolated pathogen, that is, was examined for the presence of pathogen, it was the same pathogen which was isolated from the experimentally diseased organism. So these were the 4, that is, postulates of the germ theory of disease, which were put forward by Robert Koch and which laid or became the cornerstone and are still in use to study the etiology and to study the relationship, the cause behind the disease, that is etiology, and the relationship between the disease-causing pathogen and the diseased organism, so-called the host. So the postulates of the germ theory of disease, that is, became a brought milestone advancements in the field of medical microbiology. There are some exceptions to Koch postulates, like Treponema pallidum. It's a spirochete that causes syphilis. Mycobacterium leprae, and again, a pathogen that causes a disease called leprosy, but still the that is Koch postulates are considered as cornerstones to study the etiology or the reason behind every new disease that is encountered in the field of medical microbiology. In between, Koch also perfected the technique of isolating pure culture, so-called axenic culture of a particular microorganism from a mixed population of bacteria. And he popularized the use of gelatin as a solidifying agent to prepare solid media plates for growing microorganisms under laboratory conditions. Now, this work of Robert Koch was further extended by his one assistant, Fanny Hesse. So Fanny Hesse concluded that it's not gelatin, but it is agar that can be used as a solidifying agent to prepare solid media plates for growing microbes under laboratory conditions. So Fanny Hesse had her own reasons in favor of agar, and Fanny Hesse concluded or proved that gelatin could be gets easily hydrolyzed and is not stable or suitable to be used as a solidifying agent when it is attacked by a proteolytic exoenzyme called gelatinase. And gelatin liquefies and has a very low melting point of 25 degrees Celsius. So gelatin was replaced by agar. So what were the reasons which Fanny Hesse had in support or in favor of agar? What was this agar, which was introduced as solidifying agent? It is still in use to prepare solid media and solid media plates as the most popular solidifying agent under laboratory conditions. Now, agar is actually a complex of galactose and galactopyranose carbohydrates, polysaccharides that is obtained from different species of red algae that like Gelidium, Gracilaria, etc. So what are the benefits of agar as a solidifying agent? Agar is hardly attacked by any proteolytic enzyme of bacteria and can be hardly destroyed by bacteria. And agar has a very high melting temperature of around 97 degrees Celsius and has well stable low that is solidifying points of around 40 to 45 degrees Celsius temperature. So gets well solidified at very low temperature in and around 40 or in between 40 to 45 degrees Celsius under laboratory conditions. So these were the two reasons which Fanny Hesse had in support of agar, and thus agar became the major solidifying agent to be used in microbial media for preparing that is solid culture plates for growing microorganisms. Now, there is another assistant of Robert Koch called Richard Petrie who introduced the use of Petrie plates for growing microbial cultures, and the Petrie plates are still in use for preparing for pouring media and for preparing media plates that are solid media plates for growing microbial cultures. Now, growth of medical microbiology that is progressing at a very rapid rate, and in between, important contributions were made by Edward Jenner, although they were made much before the advent of germ theory of disease by Robert Koch. So Edward Jenner had introduced already the concept of immunization or vaccination. So much before Robert Koch came up with germ theory of disease, it was Edward Jenner who perfected the technique called vaccine, or introduced the concept of vaccination. So he isolated lesions from smallpox disease and introduced into suspected organisms to protect them against cowpox. So this was the concept of vaccination. That is, the lesion from smallpox was an antigenic preparation that was introduced to stimulate immune response and in healthy organisms and to provide them protection against another disease, which was smallpox. And this was so called as vaccination. So Edward Jenner in 1798, by early 1800, had already introduced the concept of immunization or vaccination, and the word vaxa means cow, for he had used pus from smallpox and introduced it into healthy individuals to provide protection against cowpox disease. Now, another major, that is, important developments in the golden era of microbiology, that is, a series of developments were made by Lord Joseph Lister. He was an English surgeon who was impressed by the contributions made by Louis Pasteur and Robert Koch in the field of medical microbiology. So Pasteur had concluded that microbes could make human organisms sick, could make human organisms fall sick. And thereby Lord Joseph Lister, who was working on on antiseptic surgery and working over sterilization, Lord concluded, that is based on his experiments, that microbes, that wound infections are indeed caused by microorganisms. So in order to prevent such wound infections or microbes from entering wound infections, Lord Joseph Lister popularized the use of phenol as an antimicrobial agent or sterilizing agent, which was sprayed over wound dressings to prevent the entry of microorganisms. So not only this, Lord Joseph Lister also popularized a fine spray called a mist of carbolic acid that was sprayed into his operation theater before, to sterilize the air before the operation was performed. Moreover, Lord Joseph Lister soon developed or introduced the method of sterilization and started using physical and chemical agents to sterilize his surgical instruments before performing surgery at his operation theater. So Lord Joseph Lister was the first to introduce the concept of sterilization and the use of physical and chemical agents to sterilize and prevent or remove unwanted microorganisms from the surface of an object or from any object. And this was the concept of sterilization. And because of his immense contributions, he is considered as the father of antiseptic surgery. Yes, Lord Joseph Lister, because of his contributions in the golden era of microbiology, that is, who introduced the concept of sterilization and the use of physical and chemical agents to sterilize objects to make them free of microbes, is considered as the father of antiseptic surgery. The last development that took place in the golden era of microbiology was in the field of chemotherapy. Now, these were the developments. Now, the slide shows the developments, that is, the contributions of Lord Joseph Lister, who popularized the use of physical and chemical agents for the purpose of sterilization and is regarded as the father of antiseptic surgery. Now, the last development in the era of modern microbiology took place in the field of chemotherapy. And there were a lot of experiments which were conducted by Paul Ehrlich. Now, Paul Ehrlich had started experimenting with dyes to inhibit or to kill the disease-causing pathogenic microorganisms. And as a result of frequent experimentation, he found a dye called trypan red is active against the trypanosome African pathogen causing sleeping sickness. So Paul Ehrlich was the first to find the use of trypanrid in inhibiting the pathogen causing African sleeping sickness, the dye which he named or called as magic bullet. Now, this was not the end. Paul Ehrlich experiments continued, his works immense. That is, he kept on designing ingenious experiments, and in 1910, Ehrlich and his co-workers introduced the drug called salvarsan or arsenobenzol to treat syphilis that is caused by a spirochete called Treponema candida. So the introduction of the first chemotherapeutic agent or chemical called salvarsan or arsenobenzol to treat syphilis marked the beginning of the era of chemotherapy, and it was Paul Ehrlich who introduced the concept of chemotherapy by inventing arsenobenzol or the chemical or drug called salvarsan. So chemotherapy is the use of certain chemicals to kill pathogenic organisms without causing any harm to the body of the host or without causing any harm to the body of the diseased individual. So with Paul Ehrlich's work ended the golden era of microbiology, but the further developments which laid the foundation stone for the era of molecular microbiology or modern microbiology were again in the field of chemotherapy, and these developments were the advent of, that is, a series of antibiotics, one after other, being made by different biologists. We'll have a look. In between, now, developments took place in the field of virology, while the first virus particle was crystallized and isolated. This was a plant virus called TMB, and it was crystallized and it was isolated by a biologist or virologist, that is Ivanov. Further developments took place in the field of soil microbiology. Vinogradsky introduced a column called Vinogradsky column and is considered as the father of soil microbiology. Beijerinck in between introduced enrichment culture technique for selective enrichment and for selective isolation of a particular microorganism from a group of mixed population of microbes. Now we'll take a different look over these developments, that is pertaining to the other domains of microbiology like soil microbiology, virology, then that is enrichment culture technique in a different lecture session. Now let's move on to the advent of antibiotics, which marked the beginning of the era of modern microbiology. So can you name the first antibiotic which was discovered? It was penicillin that was discovered by Alexander Fleming in 1929. And therein lies a very interesting story that revolves around that discovery. Let's have a look. So Alexander Fleming in 1929 was working with gram-positive colonies of Staphylococcus aureus. He had grown Staphylococcus aureus over a plate, and accidentally the plate was contaminated by a mold or a fungus named as Penicillium notatum. Now, Alexander Fleming did not discard the plate, but kept on observing and observed that the fungus, the contaminated fungus, that is a Penicillium notatum, which had somehow or by chance contaminated the plate, was extracting a particular substance that was inhibiting or killing the colonies of Staphylococcus aureus. So he isolated the fungus, isolated the extract from the mold Penicillium notatum, and he discovered a compound called penicillin. So this was the first antibiotic which was accidentally discovered by Alexander Fleming in 1929. Penicillin belongs to a group of antibiotics called beta-lactam antibiotics, which have beta-lactam rings as a part of their structure. And the advent of penicillin, that is, marked the beginning of the era of modern or molecular microbiology. And with the advent of penicillin, the use of antibiotics came into practice for treating diseases. So Fleming concluded that there are antibiotics are substances produced by microorganisms which could inhibit the growth of other microorganisms. And penicillin in 1929 was the first antibiotic to be discovered. This was followed by the discovery of a series of other antibiotics. In 1950, Waxman and his co-workers who were working with an actinomycetes low-G plus C gram-positive bacteria called Streptomyces gracious discovered and extracted an antibiotic called streptomycin, which is used as a popular treatment regimen for treating the disease Mycobacterium tuberculosis. So next to penicillin, streptomycin was the second antibiotic that was discovered by Waxman and his co-workers. So it was obtained from Streptomyces gracious, a low G plus C gram-positive actinomycetes bacteria, and actinomycetes are low G members of actinomycetes, that is low G plus C gram-positive species, so-called like Streptomyces were, that is popular, that has focused, were popular sources of study, points of study, and became popular organisms from which a series of antibiotics were next discovered. And a series of antibiotics were soon discovered, were isolated and discovered, each and every antibiotic from a different actinomycete, like from Streptomyces venezuelae, from that is Streptomyces lavandulae, vancomycin, and other antibiotics were soon discovered. And with this, the use of antibiotics became very popular to treat or inhibit microorganisms, disease-causing or pathogenic microorganisms. Now let's summarize the developments all together that took place in the golden era of microbiology. The golden era started in 1860 and ended up in 1910 with the advent of chemotherapy and salvarsan by Paul Ehrlich, which marked the beginning of the era of chemotherapy. In the golden era of microbiology, important contributions were made by Pasteur, Louis Pasteur, who put the theory of spontaneous generation to rest and proved the doctrine of biogenesis, introduced the concept of fermentation and the role of yeast Saccharomyces cerevisiae in the fermentation of grape juice to wine, introduced the concept of pasteurization. Then next to Pasteur in the golden era of microbiology, it was Robert Koch who discovered the first pathogenic bacteria, Bacillus anthracis, the causative agent of anthrax. He next to that discovered Mycobacterium tuberculosis, the causative agent of tuberculosis. He popularized the use of gelatin as a solidifying agent that was later replaced by agar, and this was made by one of the assistant of Koch called Fanny Hesse. And the most noteworthy contributions of Koch are the cornerstones or the postulates of the germ theory of disease to study the etiology and the causal relationship between the disease-causing pathogen and the diseased individual or the host. Now, what were the other developments? Then one major development was the first use of and the introduction of physical and chemical sterilizing agents to sterilize objects and to make them free from microorganisms. That was done or introduced by Lord Joseph Lister, who is considered as the father of antiseptic surgery. Edward Jenner, before the golden era of microbiology, had already introduced the concept of vaccination and immunization, and the to conclude, the golden era of microbiology ended with the beginning of chemotherapy by Paul Ehrlich. Paul Ehrlich invented or discovered arsenobenzole, which marked the beginning of the era of chemotherapy. And after that is developments in the field of chemotherapy continued, which marked the end of the era of the golden era of microbiology and the beginning of the era of modern microbiology. And that these developments were the discovery of a series of antibiotics, the first one being penicillin, discovered by Alexander Fleming in 1929, which was isolated from a mold Penicillium notatum. Now, other developments or major developments that have taken place in the field of molecular microbiology, that is, include developments in the field of DNA sequencing, then developments, that is, Waxham, Sanger's method and Gilbert's method are the two methods that have been developed to sequence the major nucleic acid, the genetic material of organism, that is DNA, polymerase chain reaction to amplify a gene under laboratory conditions, was introduced by Carey Mullis in 1985. Then the first microbial genome sequence of Haemophilus influenzae has already been published. So a lot of developments have taken place in the era of modern microbiology or molecular microbiology, so which all of these need a very separate discussion. So next to this, that's all for today. I'll come up with a new lecture session, with a very thrilling lecture session discussing about the major developments that took place in the era of modern microbiology and paved the way, and as a result of which microbiology arose as a major discipline in this era of molecular biology. So hope you have enjoyed the lecture session. If you have really liked the lecture session, if you really, if you have truly if you find it informative and interesting, do subscribe my channel to receive more lecture sessions that is on microbiology, biochemistry, and molecular biology, meant for BSc and MSc degree courses, and for the pertaining, that is corresponding to the syllabi of various competitive examinations. In a separate lecture session, I'll be discussing very important questions that is concerned, or questions pertaining to this history and development of microbiology that have been previously asked or already asked in important competitive examinations, and that is BSc and MSc degree examinations of different universities. I'll come up with a different lecture session for that, for discussing the questions. And if you still have, after listening to this entire talk, if you still have doubts and queries, do leave your Gmail ID in the inbox to receive a free study material over the topic. Hope you have really liked and enjoyed the session. So stay tuned for the next session. Till then, goodbye.